So this chapter is all about how cells generate energy for the existence. And first we need to learn about the function of the molecule known as ATP. Okay, so the term metabolism refers to all reactions that provide chemical components for the cellular growth. So they mention energy and substances. That's essentially what it is. Energy and substances. Okay? Energy and carbon. Metabolism consists, for the most part, of two types of the reactions. The catabolic reactions, they break down big molecules. When you have starch and starch is broken down to glucose, that is a catabolic reaction. Another type of the reactions is anabolic. Anabolic reactions use energy provided by ATP to make large complex molecules. Now, metabolism can be divided roughly in three stages. The first one is digestion and hydrolysis. This is the catabolism of proteins and carbohydrates and so on when in the gut, for the most part. For example, starch is broken down to glucose, and glucose is transported from the intestines into the bloodstream. Second stage is the degradation. When you take this molecules that you just produced during digestion, and you convert them into smaller intermediates, usually consisting of 2 or 3 carbons. And finally, you oxidize the small molecules in the process called citric acid cycle in the electron transport chain. And that provides energy for ATP synthesis. In the process that you see on the screen, the large molecules are gradually broken down to effectively carbon dioxide and water at the end to produce energy. So here in this illustration you can see those 3 stages of metabolism. So first, you have digestion, right? You probably can see it. Better. So during digestion, proteins are broken down to amino acids, polysaccharides to monosaccharides, and lipids broken down releasing fatty acids and glycerol. Then amino acids, monosaccharides such as fructose, galactose, and glucose, and fatty acids are further catabolized, further broken down to carbon intermediate called acetyl-CoA. So 2 carbon compound. And this acetyl-CoA enters citric acid cycle where it's completely oxidized to carbon dioxide, eventually water. Well, citric acid cycle and electron transport chain. So the energy that you get from that oxidation is used to generate ATP. Does that make sense to you? So you can think of it in several ways. One way, you're familiar with the concept of steam engine, right? Or even better, the gas engine in a car. What is the source of energy? Gas. Gasoline, right? What happens to it? It burns. Okay? You mix gasoline, you mix air, oxygen and air, burns gasoline. Your cells basically burn large molecules, proteins, carbohydrates, fats. But your cells can't burn them at 1,000 degrees Celsius, right? They will burn them through many steps arriving to the same products, carbon dioxide and water, just it takes longer. But it can be done at, body temperature. Sounds good? Okay, let's move on. So the main structure that's responsible for metabolic processes in the cell is mitochondria. Okay. In addition to mitochondria, which is here, animal cells also have nucleus. That's where DNA is. They're surrounded by cell membrane. They filled with a gooey substance called cytosol. Okay, and there are a lot of organelles inside of the cytosol. The common question: what is the difference between a cytosol and a cytoplasm? Think about a cell as a vessel. Okay, all good? Inside of that vessel is the nucleus. The space between the nucleus and the membrane is filled with goo. The goo itself is cytosol. Am I clear? In that goo, you have other organelles, like mitochondria. The goo plus organelles is a cytoplasm. So this is what we just discussed. You must be able to match the component of the cell in its general function. Okay? But since we're going to be talking about ATP synthesis, we're largely going to be focusing on mitochondria. So, I don't know if you've heard that expression that ATP is the energy currency of the cell. You have, right? Now, the question that I don't think is asked frequently enough: why do we even need that? When I say a word currency, just currency, what does that mean? What is the currency in the United States? Huh? No, it's money. Currency is money, right? Why do we use money? I'm not joking. It's a serious question. Why do we use money? Okay. Can I ask, okay, well, the only thing that I can do relatively well is I can teach, right? But I need to feed myself, to feed my family, to pay for the housing, and so on and so forth. So imagine somebody can, I don't know, do plumbing. So I need plumbing services. I call a plumber and I go, dude, can you come over and fix whatever? And I will teach you. That sounds rather stupid, right? Because he may not need it, but another person may. So money enables us to exchange goods and services in a convenient way. Like if you call your baker and do some favor for them, they don't pay you in bread because you may not need 64 loaves. They pay you in money. Clear? Same idea is behind ATP. Your cells can use fat, carbohydrates, even proteins as energy sources. Do you follow my train of thought? You need this energy to, I don't know, make proteins. So you will need a very intricate network of biochemical reactions to say, okay, I'm going to use starch to make protein, and then I'm going to use lipids to make protein, but I need different set of enzymes. So it's much easier to have just few, like 2 or 3 main pathways to break down fats and carbs, convert it into ATP, and then plug ATP into every cellular process that requires energy, just like money. So ATP is the energy currency of the cell. It's not a storage of energy. It's the matter of exchange. It's your checking account. It's not savings. So What is ATP? You have adenosine, the nucleoside, and you can see that on the 5' carbon there are 3 phosphates. Am I making sense? So the energy is stored in the bonds between gamma and beta and alpha groups. Okay, so the most distant group, this one is, it's a gamma phosphate, that is beta phosphate, and that is alpha phosphate. Okay, so the bonds between gamma and beta and alpha are high energy bonds. So if you take a molecule of ATP, add some water and enzymes, and break off the gamma phosphate, the distant one, this breakdown will release a significant amount of energy, 7.3 kilocalories per 1 mole of ATP. You can further take this ADP and break off beta phosphate. This will also release 7.3 kilocalories. That energy can be used for, I don't know, protein synthesis, DNA synthesis, RNA synthesis, you name it. Okay, so we can describe the flow of energy in the cell the following way. Look at the illustration on the right. So you consume food. Catabolism of food, breakdown of food, releases energy. This energy is used to produce ATP. ATP is then used in reactions that are anabolic, like muscle building, synthesis, okay, as the energy source, but it can only be used as the energy source if it's broken down to adenosine diphosphate and the phosphate residue. These two can be recombined by investing energy that we receive from catabolic reaction, and the cycle goes over and over and over. Does that make sense? So now we're going to chat about first stage of catabolism, the digestion stage. And when we digest carbohydrates, enzymes break down starch, break down glycogen, producing glucose, maltose, and dextrins. Okay? So eventually carbohydrates, the breakdown of carbohydrates starts in your mouth. Did I mention that about amylase? Yeah. It starts in your mouth. Then it doesn't happen in the stomach because pH is so low and enzymes cannot function there. But then those carbohydrates enter the small intestine where pH is much closer to neutral, and effectively all ditrisaccharides are broken down to glucose. If we're talking about sugar, fructose, and if we're talking about the milk sugar, galactose, I'm sorry. Does that make sense to you? Not a great analogy, but imagine that you want to build, I don't know, a house. You have an old house that nobody uses and you can break it in pieces, right? So go there and you break the walls, the brick walls in pieces, large chunks, and you carry it in the truck and you put them on your lot. And then you break each large chunk into smaller and smaller bits until you get to individual bricks. Good? Same idea. So individual bricks would be monosaccharides such as glucose, fructose, or galactose. Okay? So these monosaccharides are absorbed into the bloodstream and they go to the liver. If it's a glucose, well, it's glucose. If it's a galactose or fructose, they will be converted to glucose in the liver. Sounds good? Cool. Now, as we mentioned, starts in the mouth and finished in the small intestine. Now, let me correct some things. The digestion of lipids does not start in the small intestine. Digestion of lipids occurs in the mouth, stomach, and small intestine. Okay? Does that make sense? So this is basically the summary of the catabolism of carbohydrates. So what happens in the mouth is that polysaccharides broken down to smaller chunks, which may consist of like 2, 3, 4 glucose units. Then when the smaller chunks end up in the small intestine, they are broken down to monosaccharides. Then they get into the bloodstream and eventually delivered into the cells in the form of glucose. So all this galactose and fructose, they will be, changed into glucose as well. Okay. Yes, lactose intolerance is for the most part genetic disorder, and people do not produce the enzyme lactase. Okay? Now, I want to clarify. So, some people have age-related lactose intolerance. And I want you to understand something else. It's a gradual thing. Do you understand what I'm trying to say? Like, when I'm a child, I produce lactose, and I break down milk sugar easily. When I become older, I may lose some of the ability to digest lactose. I may not. Like, I personally, I didn't. I can drink a couple of glasses of milk without risking exploding. Okay? Or shitting myself. But some people will have less lactase activity than I have. Will they be able to handle a little bit like a small slice of cheese? Sure. But 2 glasses of milk, probably they're going to have a lot of discomfort. And there are people who have no lactase activity at all. That's, you have to watch for, presence of lactose. Or you can just get a supplement. Okay? Supplement that contains the enzyme called lactase. And it will take care of lactose. Making sense? Now, fats. So here's the problem with a fat. Carbohydrates are water soluble, so it's easy to work with them. Lipids are not. So they have to be solubilized. They have to form micelles. So let me ask you this. Imagine you just cooked some bacon and there's grease on the skillet. Still liquid, it's warm, it's not super hot, warm, okay? You pour some water into it. What's going to happen to that grease? What is it going to look like? You have grease or oil, whatever, in the skillet, and you pour some water, you try to wash it, pour some water, what's going to happen? Are they going to mix? No. It's going to be like blobs of fat, right? Then you take your dish soap and add there. Have you ever tried it? What's going to happen to fat? It's going to get solubilized because dish soap will break it in a gazillion little bits called micelles. And then you can wash it off. It basically promotes the mixing of fat and water. Bile salts do exactly the same. Bile enters your small intestine, mixes with fat, and breaks it down into micelles. Okay? So, this is exactly what's going on when you consume fat that we know as triacylglycerols or triglycerides. So, this is your triacylglycerol, right? And you break it down, interestingly enough. 2 fatty acids. Question, lipids, are they hydrophobic or hydrophilic? Hydrophobic. So you have 2 hydrophobic fatty acids, and you have hydrophobic monoacylglycerol. You package it into micelles, as we mentioned, okay? And this products of breakdown are moved into the cells of the intestine, where they recombine back to triacylglycerols. Sounds good? What is the main component of your blood? Water. So if you will try to squeeze that fat in the blood, it's not going to go well. They don't mix. So instead of what you do, you load triacylglycerols on the protein, forming chylomicrons. If you will ask someone to eat an absurd amount of fat, I don't know if you're familiar with this phenomenal Eastern Slavic dish, cured pork fat. No? It is absolutely delicious. It's like a shot in the heart in terms of the healthiness. It is literally raw pork fat, which is cured in brine, often with garlic, and it's frozen. So it's solid. Like you take it out of the freezer, cut into pieces, put it back in the freezer. It's not supposed to be like soft and mushy. If somebody, like I love it, if I would eat a lot of that fat, the amount of fat that goes into my blood, amount of chylomicrons, is so large that if you will draw my blood, I'm not kidding, you spin it down, my plasma will be visibly cloudy because of chylomicrons. Okay? It looks quite terrifying. Right? Then it gets into mostly liver, and it's processed, being broken down to glycerol and fatty acids. Okay? All good? Question? Jackson? No? Right? Okay. So now proteins, pretty straightforward. You start breaking them down in the stomach. That is true. And proteins are denatured, and there's an enzyme called pepsin that breaks down peptide bonds. And then from stomach, those bits of proteins go to small intestine, where a bunch of other enzymes, including trypsin and chymotrypsin, break down polypeptides to amino acids. Amino acids are absorbed into the bloodstream and end up in the cells. Now, I want you to keep an hierarchy of the nutrients in your mind. Hear me out. The primary source of energy are carbohydrates. the secondary and most efficient source in terms of like energy density. Lipids, fats. Proteins are used for energy. That's like a third order, because for the most part you need amino acids to build tissues. Is that clear? So, in the process of energy generation, we oxidize, we strip molecules of electrons. The problem is those electrons have to be carried from one point in the cell to another. Electrons cannot exist by themselves, so the electron carriers, such as NAD plus and FAD, serve as coenzymes. Okay? What to explain? Okay, hear me out. You have an enzyme that is supposed to do something, right? It cannot do it without coenzyme. It is required for the enzyme to function. So let's figure out how they are required. A very quick crash course in oxidation and reduction. So when a reaction is a reaction of oxidation, look what it involves. Listen, a loss of hydrogen or loss of electrons, or the increase in the number of bonds that connect carbon or other atoms to oxygen. Reduction reactions include quite the opposite. The gain of hydrogens, the gain of electrons, or the decrease in the number of bonds to oxygen. So this table summarizes the difference between oxidation and reduction. Here's what I want you to understand. When you oxidize something, the energy is released. When you reduce something, you put energy in. Am I making sense? So let me ask you this. When you put gasoline in the tank of your car and you burn it in the engine, is energy released? Yes. So your engine oxidizes gasoline. Well, oxygen in your engine oxidizes gasoline. Am I making sense? Just keep it in mind. So the first coenzyme that we're going to mention is NAD+. Now, remember, let me go back for a second. So, take a look at the first column in this table. Oxidation involves the loss of electrons and hydrogens. Do you agree with me? I just stated what's written, all right? All good? Cool? Let me ask you a profoundly stupid question. When the table says that it includes the loss of electrons, does it mean that electrons completely disappear? Why? You do. Fundamental law of nature. Law of preservation of the M word. Everything that surrounds us is what? Matter. Mass matter, yes. So, the law postulates that matter isn't created or destroyed. Electrons are the part of matter, right? Does that make sense? So, electrons aren't lost forever. They are stripped off, right? but they have to be carried to something else. Something has to accept the electrons. When you burn gasoline in the car, electrons are accepted by oxygen. So it's not a problem to carry electrons in the engine because everything is mixed, but in the cell, you can have one reaction like here and another reaction there. So you have to carry those electrons and hydrogens from one place to another. This is the function of NAD+. It sometimes is called an electron carrier. Think of it as a taxi for electrons. So NAD+ is called an oxidized form when it accepts, remember, when it gains electrons or hydrogens, it becomes NADH. And when it gains electrons, it is reduced. Do you follow? That's a definition. So NADH is a reduced form. When NADH loses electrons and hydrogens, it goes back to NAD+. It becomes oxidized form because the loss of oxygen and hydrogens is oxidation. Okay? So NAD+, look at another example. So you have ethanol, right? When ethanol is oxidized, how many bonds between the oxygen and the carbon in the molecule of ethanol? How many bonds? Oxygen and carbon, the one that I highlighted. 1. How many bonds between oxygen and carbon in the molecule? 2. So you increase the number of bonds to oxygen, which means you oxidize. Does that make sense? What you get is a reduced form of NADH that carries electrons. Now what you see here, alcohol dehydrogenase reaction, is the first reaction of alcohol metabolism. Like when you drink alcohol, this is what happens. Okay? Clear? So it generates some electrons. We're going to get back to this idea later. Another coenzyme with a very, very similar function is FAD. So, same story. You have oxidized form, FAD, which can accept hydrogen and electrons, becoming a reduced form, FADH2. Okay? FADH2 can lose hydrogen and electrons, going back to its FAD form. Good? So FAD also participates in the reactions that oxidize organic compounds. However, its main function, check this out, not the bonds between the carbon and oxygen, but bonds between two carbons. So take a look at succinate on the left. How many hydrogens that I highlight do you see? 4. Look at the fumarate on the right. How many hydrogens that I highlight do you see? So we lost 2 hydrogens. The loss of 2 hydrogens is oxidation. Succinate was oxidized and produced fumarate. Okay? Those hydrogens got accepted by FADH together with electrons. Any questions right now? All right. Now, I, quite frankly, have no idea why anyone wants to memorize the structure of coenzyme A. I definitely don't want to. You have ADP, you have pantothenic acid, aminoxanthiol. There's something there I want to highlight that's common between all these 3 coenzymes. I'm going to make a big highlight of pantothenic acid, right? For FAD, riboflavin, vitamin B2. For NAD+, it contains niacin, vitamin B3. Okay? My point that I want to make. The role of vitamins in human body. They are essential for the enzyme function. They provide the source for those coenzymes. Am I making sense? Okay? So the function of coenzyme A is that it can transfer so-called acetyl group onto some other organic compounds. Okay? So it can carry the acetyl group from one location in the cell into another. You may have noticed the common pattern between coenzyme A, NAD+, and FAD. Coenzyme A, listen, carries acetyl group. FAD carries hydrogens and electrons. NAD+ carries hydrogens and electrons. They all carry something. So now that we are equipped with this knowledge, we're going to talk about the whole process of the glucose oxidation. So the very first step of glucose oxidation is called glycolysis. Glyco comes from glucose, and lysis is a breakdown. Okay? What I think we can achieve today, hear me out. We're going to go through glycolysis, Krebs cycle, and the electron transport chain, and that probably should be enough. So we can leave lipids and proteins for Monday next week. Okay? So, what does glycolysis do? It takes a 6-carbon glucose and breaks it down to 3-carbon pyruvate. Glycolysis is always in the cytoplasm, outside of any other organelle, and it does not require any oxygen. In terms of evolution, it is one of the oldest, if not the oldest, metabolic pathway ever evolved. All cells, and I'm not making this up, all cells, from bacteria to humans, in some form, with some modifications, carry out glycolysis. So glycolysis consists of a whole bunch of reactions, 10 in this example, but there are two main phases. The one that I highlight here is called energy investment phase. You can ask, wait a minute, why is it energy investment? Well, I want you to take a look at a couple of steps. Remember how this book shows you ATP. Do you see this ATP here? The top is ATP. The bottom is ADP. When you convert ATP to ADP, when you break off one phosphate, is energy released or is it used? When you break down a high energy bond, released. This is why it's energy investment phase. That makes sense? Do we see the first highlighted ATP? All good? Second highlighted ATP to ADP transition, can you see that? So during energy investment phase, we take two ATPs and use their energy to produce this high energy intermediate called glyceraldehyde 3-phosphate. Okay? All good? So this illustrates the first investment, okay, which results in phosphorylation of glucose. And then glucose is isomerized. And then you have the fructose phosphate. And then fructose phosphate receives another phosphorus in the second investment reaction. So you spend a second ATP molecule. So far, so good? This fructose 1,6-biphosphate is broken down to two different carbonyl group containing molecules. On the right, you see glyceraldehyde 3-phosphate. On the left, you see dihydroxyacetone. Okay? These two different molecules, the one on the left is isomerized. So all in all, at the end of energy investment phase, you are stuck with glyceraldehyde 3-phosphate. Okay? Sounds good? Now, if you go back to this scheme, do you see the glucose in the very beginning? Carbon atoms are represented as black circles. How many carbon atoms do you have in glucose? 6. So this is your intermediate glyceraldehyde 3-phosphate. How many carbon atoms do you have in each molecule? How many molecules do you have in total? 2. So how many carbon atoms do you have there? Total, in 2 molecules. 6. You start with 6 carbons in glucose. You finish in your investment with still 6 carbons. No carbons have left the reaction. All good? And then you have energy payoff, or energy generating phase. So glyceraldehyde 3-phosphate is phosphorylated by inorganic phosphate and dehydrogenated, which generates NADH. Remember, does anyone remember what they carry, the NADH molecules? What they carry. They are carriers. What do they carry? And something else? Hydrogens. Yeah. Electrons and hydrogen. Very good ones. Very good. Then, this molecule that we generated previously, 5-biphosphoglycerate, is donating phosphorus to make a molecule of ATP. Okay? And then phosphoglyceric acid forms what's called phosphoenol pyruvate, and phosphoenol pyruvate generates another molecule of ATP, resulting in pyruvate. Now, there's no intention to put any of these intermediates, except for maybe the middle one, onto the exam questions. So I want to review this energy generating phase again, focusing on the number of carbons. How many carbons do you have in glyceraldehyde 3-phosphate? 3. How many carbons do you have in the pyruvate? 3. Did we lose any carbons? We didn't. One more question. I'm going to scroll all the way back up here. I don't have enough. How many molecules of glyceraldehyde 3-phosphate do you have at the stage 5? 2 3-carbon molecules. can you count to 2? Can you count to 4? It's a rhetorical question. So let's take a look at the energy generating steps. How many, hear me out, how many NADH molecules are generated in energy generating phase per 1 glyceraldehyde 3-phosphate? How many NADH molecules do you see on the screen? 1. 1 NADH per 1 glyceraldehyde 3-phosphate. Everybody agrees? How many molecules of glyceraldehyde 3-phosphate do we have? 2. 1 molecule gives you 1 NADH. 2 molecules give you 2 NADH. All good? Now, does that logic make sense? Keep counting. How many ATPs do we generate in this reaction? How many ATPs do we generate in this reaction? 1. These 2 reactions, how many ATPs do they give us? 2. These 2 ATPs are generated per 1 molecule of glyceraldehyde 3-phosphate. Do you agree with me? I'm going to ask again. So 2 ATPs per 1 glyceraldehyde 3-phosphate. How many ATPs are we going to get for 2 glyceraldehyde 3-phosphates? 4. Now, this is the summary. Well, sorry. To give us that summary, I'm going to go back to that scheme and take a look at it again. How many ATPs do we spend during the energy investment phase? 2. Everybody sees them? 1, 2. How many ATPs do we get back during energy generating phase? 2 and another 2, right? 4 total ATPs are produced during energy generating phase. Do you see that? If not, tell me now. Let me know. I'm going to circle them. So, no, not there. Let me clear everything because it's a bit crowded and we're going to go over this. Okay. I'm literally going to ask you to count maximum to 6. And I'm going to talk to you, okay? So first I want you to look at the energy investment phase. Sorry, I'm going to single Sue out and I'm going to talk to her. And also, my bad, I'm going to stop it for a second. Okay, let's start with energy investment, shall we? The system a second, it's kind of stupid. And yeah, I think this tablet is like me, underslept and overworked at this point. Oh, Jesus Christ. I can just do something about it. Okay. Well, the system decides whether is going to cooperate with me or not. I want you to take a look at the first step of energy investing phase. How many ATP molecules do we have? No. First step. How many molecules we use? 1. Second step. How many molecules we use? Sorry, third step, how many molecules we use? So what's the total? Keep it in mind, will you? So, use one, okay? Then we produce 2 molecules of glyceraldehyde 3-phosphate. Agree with me? Each molecule of glyceraldehyde 3-phosphate generates 2 ATPs, right? So 2 molecules will generate how many? Total. 2 in one reaction, because it's 2 molecules, and 2 in another reaction. So what's the total generation? 4. So you generated 4 ATPs. How many did you spend in the beginning? You gained 2. You gained 4. You spent 2. What's the net yield? 2. Do you understand why? You invested $2, you got back $4. What's your profit? 2. You invested 2 ATPs, you got back 4 ATPs. What's your ATP profit? 2. Does that make sense? So glycolysis net yield is 2 molecules of ATP and 2 molecules of NADH. Here. Okay, cool. And at the end of the day, you have pyruvate, also 2 molecules of pyruvate. We got this? Okay. So first of all, not impressive, really. Not impressive. The yield is poor, right? You got through like 11 steps. And what you get? Just 2 ATPs. It's a pretty meager yield. Well, you also get 2 NADH, but what's important? You get 2 pyruvates. All good? Pyruvate has 2 options. There's no, and we're talking about human cells. There's no oxygen if cells do not receive oxygen. Anaerobic conditions. Pyruvate will get back the electrons from NADH, electrons and hydrogen. It will be reduced. It will gain electrons and hydrogens, and it will be reduced to lactate or lactic acid. Sounds good? If oxygen is present, though, pyruvate will get into mitochondria, where it will be oxidized even more, losing electrons and being converted to acetyl group, which is carried by coenzyme A, so-called acetyl-CoA. Am I making sense? Here's a very important question, and I'm not kidding. How many carbons are in the pyruvate? How many carbons in this acetyl group? Can carbons disappear completely? No. The preservation of matter, right? So this carbon, the difference in carbons, should go somewhere. This carbon that we're losing it in the form of carbon dioxide, CO2. All good? Okay. So let's take a look what happens in those aerobic conditions. So we already figured that this 3-carbon pyruvate gets into mitochondria and gets oxidized. So it loses electrons and hydrogens. You probably have noticed sort of a common theme. When it loses electrons and hydrogens, where do they go? The answer is literally written on that slide. Which molecule accepts electrons and hydrogens? NAD, becoming NADH. That's your electrons and hydrogens. Okay? How many carbons are in the pyruvate? 3. How many carbons in the acetyl group? 2. So the third carbon escapes in the form of CO2. All good? Now we have acetyl-CoA. It's going to go into Krebs cycle, but let's take a look what happens in anaerobic conditions. I really want to talk to authors of that book and tell them, can you please stay in the field that you know, which is chemistry, not biology. So, we saw that in anaerobic conditions, pyruvate is converted to lactate. Okay? This part. Right? Now you can see what happens to NADH. Tell me, please. NADH loses hydrogens and electrons and becomes what? NAD+. And where does NAD+ go? Back to glycolysis when it accepts electrons and hydrogens and becomes NADH. You see what I'm saying? NADH is a full taxi. It's a taxi with passengers. Passengers are hydrogen and electrons, right? NAD+ is the empty cab. You follow? Now up to that point, everything's right. Everything is right. But strenuous exercise. Why your muscles, especially when you do like heavy lifting resistance training, they work under anaerobic conditions. Why do you think it is so? It's much less true for things like walking or running, jogging. Why are your muscles anaerobic? It's a hard question. Yeah, but why? What carries oxygen around the body? Say again? No. Like why? So what carries oxygen around the body? Where does it take oxygen from? Lungs. So now you know this. Blood carries oxygen around body, right? Why don't your muscles get enough oxygen with the blood? Huh? Say again? Not quite, but it's actually a correct thought. Or like it is relevant thought. So let's break it down. Your muscles, do they receive blood? Why don't they receive adequate amount of blood? This is a really hard question. Especially when you lift heavy. What do you need to do with your muscles to lift heavy discs off of the floor? Contract. Right? Shorten. What happens to your muscles in terms of the width, the volume, when you contract them? They become what? Contract a muscle. What happens to them? They expand, they bulge. What do you think happens to blood vessels within that bulging muscle? They squeezed. Can blood go through those squeezed blood vessels? Not sufficiently, no. So effectively, when muscles bulge, they squeeze blood vessels, and there is an inadequate blood supply to the muscle. Am I making sense? So it's not your lungs. Your lungs provide enough oxygen. So your muscles become anaerobic. What do they accumulate? Lactate. Then becomes a turn of bullshit statements. Well, not entirely bullshit. First of all, imagine that you work really hard and immediately after the exercise your muscles kind of have the burn feeling. You know what I'm talking about? Huh? Say again? No, no. We'll get to this, not DOMS. Immediately after, it kind of burns. It is somewhat a satisfying feeling. Okay? I can tell you that lactate that accumulates in your muscles is gone in an hour. So it does contribute to that initial sensation of soreness, but it's a short-lived soreness. Lactate accumulation has nothing to do with fatigue, with the tiredness, because it's gone. DOMS that you refer to, like next day soreness, this is the damage, good damage, to the connective tissue within the muscle. This is why it hurts next day. Am I making sense? And this soreness actually stimulates the muscle growth. Lactate-mediated soreness is very quick, very short-lived, and has nothing to do with muscle growth. Okay? I'm telling you, I can give you the evidence. Just remember that. Okay? Now, this is true. We form oxygen debt. And we generate more oxygen debt if we lift heavy. Sounds good? So when you go and do the resistance training, and then you leave the gym, all this oxygen debt, all this anaerobic metabolism, all the debt has to be repaid. So you consume more oxygen, and your metabolic rate is increased even after the exercise. I'm trying to connect all those dots to link the weight loss and the exercise. I'm going to ask you a series of questions, and I want you to try and answer them, scientific questions. Can a person lose weight only by exercising? You take a person and tell them, diet, you eat as you want, like an animal. Okay? You want to eat, you eat. You're going to work out a lot. Is this person going to lose weight? No. The answer is no. Because the hormones that are produced as the result of exercise, glucocorticoids, glucagon, and so on, they stimulate appetite so that food consumption is way over the calories spent. Am I making sense? So if a person wants to lose fat, dietary restriction is essential, or dietary modifications are essential. Is that clear? Second question, which exercise is more efficient in terms of the weight loss? Resistance training, like high load, high intensity, short bouts, basically lifting weights, or walking or jogging? Lifting weights. It creates greater oxygen debt and increases metabolic rate much more significantly than running. As much as I love running, I fully acknowledge that in terms of the overall health benefits, resistance training way outpaces running. No pun intended. Okay? That makes sense.
So is it that those who lifts are more heavier than the runners?
Diet.
Just because of looking for them.
Again, diet. Depends on it. Here's the thing. It's a good question. Why people who lift weights are bulkier than people who run. Okay. If you compare Olympic athletes, sure. Because the goal of a weightlifter is to lift as much as they can, so they need a lot of special type of muscle fibers, the type 2 fibers, the fast glycolytic fibers, to produce an effort. If you ask a weightlifter to run a mile for endurance, they're going to die. Okay? On the contrary, people who run, they need endurance, They need type 1 fibers, slow oxidative, which are small, don't lead to hypertrophy. So look at any marathon runner. They're super slim. Does that make sense? Now, if you decide to pick up weightlifting, you can adjust your diet so you don't bulk. You can be fairly strong without hypertrophy. Am I making sense? Now, to be honest, I'm not the thinnest guy in the world. But if you will ask me, and I've been running for many, many years, I'm not running competitively like, first places in marathons and whatnot. But if you ask me to run a half marathon, I will need different shoes, but I can do it like from the get-go. I built a lot of endurance. My strength, on the other hand, isn't that great. Does that make sense? So it's all about diet and exercise combination. So let's take a look. Okay, we're probably going to go over citric acid cycle and cut it short. Okay. Yeah, we're in the middle. Should produced during anaerobic conditions. Lactate. Reactions that convert glucose to pyruvate. Glycolysis. Reactions that break down large molecules to smaller molecules. Catabolic. And substances that remove or add hydrogen and oxidation and reduction are coenzymes. Let me see how much of a citric acid cycle we have. Oh, shit. Well, let's go over this as much as we can. So we're going to talk about citric acid cycle. This is the schematic representation of it. So basically the goal is to finally break down acetyl-CoA and release energy. So it uses the acetyl group, an acetyl-CoA, to produce carbon dioxide, reduced form NADH, and reduced form FADH2. The names are numerous Krebs cycle, tricarboxylic acid cycle, or citric acid cycle, because citric acid is formed in the first reaction. So basically, here's an idea. You take 2 carbons from the acetyl-CoA. You take 4 carbons of the oxaloacetate. 2 plus 4 equals 6. That's citrate. Then you remove 2 carbons from the citrate. When you remove 2 carbons from 6, how many do you have left? 4. So end up with 4 carbon succinyl-CoA. Then you have the succinyl-CoA converted in a series of reactions to oxaloacetate, no loss of carbons. So again, 4 carbon oxaloacetate, and the cycle starts over. Making sense? I'm not going to spend time on this because we're going to go over each reaction. And I could scare you and tell you need to memorize each reaction, but this is bullshit. No, I want you to learn conceptually. So let's take a look at the first reaction. All I want you to know right now is the carbon number. Okay, good? Number of carbons in acetyl-CoA, 2. Oxaloacetate, 4. 2 plus 4 equals, so this is your citrate with 6 carbons. All good? Second reaction is isomerization. How many carbons can you count in this molecule? Ooh. 6. 6 in the citrate, 6 in the isocitrate. Those are 2 isomers. Now, remind me, how many carbons do you have in isocitrate? Look at this alpha-ketoglutarate. How many carbons do you see? 5. So we lose 1 carbon in the form of CO2, but we produce NADH. What does NADH carry? Hydrogens and electrons. So that was the whole idea, to strip some hydrogens and electrons from citrate, or isocitrate. Keep in mind that alpha-ketoglutarate has 5 carbons. This is succinyl-CoA. How many carbons? 4. Where does that carbon go? CO2. We have another NADH. So another set of hydrogens and electrons. Am I making sense? Succinyl-CoA is broken down to succinate. Still 4 carbons, nothing. Number of carbons doesn't change. This is the step when Krebs cycle generates a molecule of ATP. Okay? I want you to put this molecule of ATP on the back burner. Just remember, we got 1 molecule of ATP, right? All good? Now remember that when you go from no double bonds to a double bond, it means that you lost hydrogens. When you lose hydrogens, it's oxidation. Remember that? So succinate is oxidized to fumarate. This hydrogen that it loses goes to what? FAD to form FADH2. Again, hydrogens and electrons. How many carbons are in the fumarate? 4. Malate, still 4. But you add water, so hydration. So you go from fumarate to malate. And then malate, with 4 carbons, is oxidized. You see you had 1 carbon-oxygen bond. Here you have 2 carbon-oxygen bonds. See that? Now, malate is oxidized to oxaloacetate, and the reaction starts again. You also generate an NADH. Okay? This is the overall reaction. So I want to focus on the yield on the right. So we've got 3 NADH, FADH2, and GTP, which will be converted to ATP, right? And 2 CO2 molecules. Okay? So, 1 turn of citric acid cycle. How many CO2s are produced? 2. NADH? 3. FADH2? GTP? Acetyl-CoA? 1. Why do we use 1 turn? I'm going to go all the way back here. Like seriously back. Don't worry. Glycolysis, right? How many glucose molecules do we have in the beginning? I'm not asking hard questions. How many glucose molecules do you see in the beginning? 1. How many carbons? How many pyruvate molecules do you see at the end? 2. How many total carbons? 6. Everybody follows me? So instead of 1 6-carbon molecule, we have 2 3-carbon molecules. Hang on. How many acetyl-CoA molecules are produced from 1 pyruvate? 1. 1 pyruvate on the left, 1 acetyl-CoA on the right. You see that? How many pyruvates were generated in glycolysis? I just asked you that. 2. So how many acetyl-CoAs we will produce per molecule of glucose, per 2 pyruvates? 2. So 1 molecule of glucose will give you 2 pyruvates and 2 acetyl-CoAs. Does that make sense? How many turns of the cycle do you need for 1